to the second Biology 121 online video. We're going to talk today about the process of photosynthesis, looking specifically at ATP synthase in a greater level of detail. This is going to be a relatively short video following up from an unusually long one last time, so let's dive in. Once again, we talked about photosynthesis as the opposite of respiration, so this follows up from the last video with 4 steps. First, light is absorbed, then there's an electron transport chain, ATP is formed as well, and then all 3 of those steps form what we call the light-dependent reactions. It starts with light absorption and ends with charging up 2 high-energy molecules, NADPH and ATP. Once that chemical energy is created out of that solar energy, the solar energy is converted to chemical storage. Then there's going to be a step called carbon fixation that involves a Calvin cycle and a couple of different metabolic pathways that we're going to talk about next week. For today, we're just going to focus on that ATP formation portion most intently. So this 2-step process with the electron transport chain and the ATP formation, they work really closely together, and I want to spend a little time today unpacking how they relate to each other. So here again we see a plasma membrane down the middle of your screen, and on the top of that drawing we have the stroma, which is outside of the thylakoid and inside the chloroplast. And the bottom part of that we have, it says in the lower right corner, thylakoid lumen, the interior of the thylakoid. So the line down the middle represents the thylakoid membrane and all of these different proteins that are embedded in the thylakoid membrane. So the question I'm raising is, since ATP synthase, which is an enzyme, we can tell by the ASE ending of that, is going to be charging up ATP from ADP, and pause for a moment and go find the little orange ovals that say ADP and ATP and how there's an arrow connecting them together so we can see that we're charging up that ATP, giving it energy, and also connecting an extra phosphate group onto the ADP. The question I'm raising here is, where does the ATP synthase get its energy from? We know it's not directly getting energy from light because the light is striking photosystem 2 and then photosystem 1, and that's charging up electrons, but those electrons are not directly connecting over to that ATP synthase. If we follow the flow of the arrows here, we've got light striking photosystem 2, and then an electron gets ejected out the bottom, and another electron a high energy electron gets ejected out the top. And we've got this electron transport chain in purple through the middle there, and then we've got more light striking a different photosystem, charging up a different electron, and that electron is being used to charge up in a smaller electron transport chain to charge up NADPH. But what is providing the energy for the ATP synthase itself? So let's focus just on that ATP synthase piece. We can see the lights coming in elsewhere in the process. We have an NADPH, a high energy atom, kind of sitting right there next to that ATP synthase. That's not actually part of it. That's ADPH. NADPH is going to go wandering off into the stroma and be used elsewhere in the next step when we get to the light independent reactions. But I want to suggest that you look at is that there's more of those H pluses down at the bottom of the screen inside the thylakoid lumen than there are up at the top of the screen. In fact, if we count them out across the top, the author of this diagram has drawn across the top in the stroma section 1, 2, 3 H plus ions, and you'll notice that one of them actually, the first one has an arrow pointing in to the thylakoid, and the last one is at the end of an arrow pointing out of the thylakoid. So we have kind of a pumping action where we have one hydrogen ion being brought in and one being released, but also on the very far left, down at the bottom, you'll see that H2O, water, being split open into half of an oxygen. So for every 1 water molecule, for every 2 water molecules, you get 1 oxygen atom, or oxygen molecule, and 2 becomes 4 if we got 2 waters, of that H plus again. So there's actually 1, 2, 3, 4, 5, 6, 7, 8 of those hydrogen ions inside the thylakoid lumen. So here's what's happening that gives that ATP synthase its energy. I'm going to change and do a little metaphor for you here. You may have done this in physics class, maybe last year, where you tied a balloon to a straw and then ran a string through that straw. And when the balloon is pressurized, when it's full of air, the law of diffusion, like we've talked about in our class, makes that air want to leave, to spread out as evenly as possible in a depressurized manner. And so the pressure inside the balloon can be turned into an energy that will make the balloon move. Well, we have a similar process happening inside this thylakoid, because what's happening is that electron transport chain, the B6F group, the first purple blob in the drawing at the bottom, is bringing H plus protons, hydrogen ions, into the thylakoid, and when it pumps them in there, it builds up a pressure, just like the pressure inside the balloon. Now, the balloon is full of air, which is mostly nitrogen, and here the thylakoid is getting filled up and pressurized with hydrogen ions, which is a different chemical, but the same principle basically applies. So that electron transport chain that I've just circled and labeled in the drawing is pumping ions into the thylakoid so that there's a lot of those ions stuck inside that thylakoid. Then what that does is the ATP synthase has the ability to release that pressure. And it's the release of that pressure that, just like the balloon on the string, is going to allow the ATP synthase to do the work of binding that third phosphate group to an ADP, making it into ATP, the higher energy version. So that release of pressure is harnessed as a kind of chemical energy in the form of ATP. That's going to wrap it up for today. We'll come back next week and we'll talk about what plant cells then do with all of that ATP. And the reason why they need to do something with it is because ATP, for all that's a wonderful, wonderful tool that all life on Earth uses for all of our metabolic processes. It's the source of energy we all use in every metabolic process. Anytime a living thing needs energy, it uses ATP to provide that energy, but ATP does not have a very long shelf life. If it's just lying around inside a cell for more than a few hours, it starts to go bad. It degrades. If you've ever had a battery, a rechargeable battery that just like dies on its own and is just sitting there not doing anything, it's the same exact process. The chemical will break down spontaneously. And so plants are going to take that energy from the ATP and they're going to store it in a much more stable form called sugar.